Welcome to the Honeybee Watch Symposia series. I'm Steve Rogenstein, based in Berlin, Germany. Honeybee Watch's project director and your host today. And our co-host is Arrigo Moro, the science director of Honeybee Watch. So to introduce today's symposium, it's titled, Are Wild Honeybees Wild Bees Too? And some of you find that to be a very obvious question, very rhetorical, but there's controversy around this, and that's something that we hope to discuss and explore today. So with all of our topics, we try to have our finger on the pulse and explore issues relevant to our field of study, namely around free-living honeybees, and really kind of address these topics that colleagues are talking about or are challenged by. For this one in particular, we wanted to confront an issue head-on and not shrink from its potential controversy. And that is the notion that honeybees are competing with other bee species for resources, namely nectar and pollen. I see, Rebecca, you nodding your head, and you know all about this with the mellifera in Yucatan. So as we dove deeper into this issue, its complexity became really clear. So intertwined with the issue of competition are these other kind of sub-issues of native versus alien, the degree of honeybees' invasiveness in different geographic settings. Jamie is representing the Americas. We have Kit, who's representing Australia. These are lands that are not the original geographic range of Apis mellifera. So they are, non-native, invasive, call it what you may, but that's not where they came from. So there's a level of kind of controversy there. And then also another one of these sub-issues is their effects on other species. So in this 2-hour symposium, obviously, we're not going to get enough time to dive deep into all of these subtopics and try to unpack all these layers. It's not going to happen. So what we'll do, though, is with these 3 experts, we hope that they represent a diversity of perspectives, expertise, and opinions. And through especially the panel discussion, we hope to ask questions that really target the issues. So Marija Tanasković is a senior research associate and the head of the Department of Genetics and Ecogenotoxicology at the University of Belgrade in Serbia. We were first introduced to her work through a colleague and one of Honeybee Watch's core team members, Jovana, who I think is with us still. And then together, Jovana, Marija, and others from her lab, they sampled and sequenced the genomes of countless honeybees throughout Serbia in an effort to compare the genotypes, to try to differentiate wild versus managed colonies and glean other information. She is the only speaker of our 3 who represent Apis mellifera in their original geographic range, and where we can truly call them wild, if they are wild and meet other criteria. And thus, if their genetics can be proven to come from these self-sustaining populations and not feral escapees from managed apiaries, that's how we would define them as being wild. But it's a difficult challenge, and one that Honeybee Watch is trying to address in our ongoing research. And because she is a geneticist, she has been very definitive in determining differences between these types of populations, managed and wild. So not wanting to take up more of your time, I'm going to give you 14 minutes on our clock. Is your presentation ready? Certainly. Great. Would you like to start? And I'll interrupt you when you have 1 minute left.
Thank you, Steve.
Thank you,
Hello,
Marija.
and good day to everyone. It's a great pleasure to be the part of this symposium. Thank you again for inviting me. As Steve said, I'm Marija Tanasković from the Institute for Biological Research Sinisa Stanković at Belgrade. And I'm going to tell you a little bit something about Serbian honeybees population. But first, something's wrong. My background story is that I am population geneticist. which basically means that my scientific area of expertise are not bees. What I did study throughout my whole adult life is genetic variability of different species and how gene pools are shaped by the evolutionary and ecological history these population experiences. I'm trying to find genetic differences between populations and understand how these differences help populations to adapt, cope, and survive in changing environments. I have yet a lot to learn, but I am certain that preserving genetic variability through preserving locally adapted population is extremely important for conservation of biodiversity. So we wrote the project, named it SERBHive, and got finances from the Serbian Science Fund. We dug through the literature and learned that in Serbia we have two subspecies of Apis mellifera with pretty neat geographic distribution. You have Carnica bees in north, Macedonica bees in south, and mixed population in the middle of the country. And there are also three described ecotypes of Carnica bees, which live in geographically and ecologically different areas. Our project main goal was actually to describe and assess genetic variability of all types of honeybees societies in Serbia. And since the Serbia is in the native area of distribution of Apis mellifera, we expected to find wild, feral, and managed honeybees, which is maybe the main difference between Serbia compared to Australia and America. We expected to find wild native honeybees in hard-to-reach places in wilderness, especially in national parks and nature reserves. And we wanted to answer these two questions. Are there wild honeybees in Serbia? And are there genetic differences between honeybees from different parts of Serbia that are ecologically and geographically different? Our plan was to go to that hard-to-reach places in nature throughout Serbia and do the beelining and also to collect samples from stationary apiaries and then go back to the lab and do stuff, which means hundreds of hours of bench work doing population genetic analysis. And we chose these four markers. First, a specific region of mitochondrial DNA that enables us to distinguish Macedonica and Carnica subspecies. Second is the most commonly used sequence of cytochrome oxidase 1 subunit, also on mitochondrial DNA, and that enables us to compare our results with previous findings. We were lucky to be able to do the whole genome sequencing. And till now, we assembled this 14.5 kilobase long mitogenome, but still have terabytes of data to analyze it yet, so stay tuned. And finally, to analyze nuclear markers, which are 14 microsatellite loci. We went to the field and didn't have any trouble to find apiaries, since they are everywhere, and I mean literally everywhere. But we did have some difficulties to find apiaries that met our criteria, and that criteria was that apiaries needed to be at least 10 years old, and that needed to have a minimal or no queen import, and preferably be managed in traditional way. During our field work, we discovered that beelining is extremely frustrating. And though the method works, it really works. In most cases, bees led us straight to the apiaries due to the aforementioned fact that apiaries are everywhere. And then we became a little bit desperate because our sample of bees caught in the nature was depressingly small. But then this amazing person, Jelena Bila-Dubović from Faculty of Biology, told us that we actually have a big, thriving, free-living honeybees community in Serbian capital Belgrade, where we sampled 40 free-living honeybees communities. And so what molecular genetic analysis told us is, in brief, that there are a lot of changes in Serbian honeybee populations compared to the literature data we were able to obtain. First is that we were not able to confirm presence of Apis mellifera macedonica subspecies, and this was something alarming. And that was my first experience as biologist to see decline of biodiversity and loss of genetic diversity in real life. Secondly, we detected 21 mitochondrial haplotypes, of which 10 are newly described for us. But despite that new haplotypes, all haplotypes are randomly distributed throughout the different regions of Serbia. And we couldn't see any signs of differentiation between populations or between different types of societies. Then near complete mitogenome sequences enable us higher resolution on this type of genetic markers. And again, we have the similar story. We don't see region-specific clustering of lineages, and majority of the branches contains lineages from different regions. Basically, all our analysis on mitochondrial genome showed us that there are no genetic differences between different populations in Serbia, and that we cannot see that there are differences between managed and free-living colonies that we sampled. However, our analysis actually showed us that free-living colonies have higher genetic diversity compared to managed ones. All the parameters of genetic diversity that we were able to measure show that they're higher in free-living colonies. And our microsatellite data revealed that honeybee population in Serbia is mainly admixed. Again, meaning that there are no genetic differences between populations from extremely different natural habitats. With that, we also discovered some fingerprints of Italian honeybee presence, and that is something that is not allowed by law, since in Serbia, law allows only for Canolian bees to be kept in apiaries. So we detected some import of non-native honeybees subspecies in Serbia. Also, we were happy to observe this gray area of haplotypes, which are population of Tara cyanica, and we know that there are one of previously described ecotype there. So basically, we are hoping that some part of locally adapted population is preserved in Serbia. And also, you may not see in this wood of admixture, but this green circle and this blue circle are our free-living colonies. Green are from all over the Serbia, and blue are from Belgrade. And also we can see glimpses of differentiation between free-living and managed honeybees societies. And again, all parameters of genetic diversity were higher in free-living colonies compared to all managed ones. And finally, the paper that Steve mentioned are Belgrade urban honeybees. So what we did with Jovana help, we sampled honeybees from free-living societies in Belgrade and from apiaries in Belgrade that live pretty near to each other. And here in this haplotype network, you can see that most common haplotypes are actually shared between different types of societies, but rare haplotypes are actually distinct to either managed or either free-living societies. Also, this microsatellite Lucy and this again, blue is Belgrade free-living honeybees, you can see that they are distinct from the Belgrade managed colonies that you cannot see from this wood of all other managed societies in Serbia. And moreover, free-living honeybees from Belgrade are more related to each other than to managed honeybees in Belgrade, which is a sign that they are more frequently breed between themselves. So they choose to breed feral with feral or free-living with free-living, and managed breeding is controlled by human. So what are the exactly answers to our two questions? Well, we cannot say with certainty that bees from the nature are wild according to biological definition. We don't see genetics distinctness that will be able to confirm that. But we know that free-living honeybees does exist in Serbia, only not where we expect them. And for the second question about the genetic differences between honeybees, well, answers are pretty disturbing since we could not detect Macedonian bees in our sample, and that indicate that they are not present in Serbia anymore. And this is in accordance with Serbian law, which changed some 10 years ago. And although it appears that Serbian beekeepers are law-abiding citizens, that changes in government practice without consulting experts from different areas can lead to really detrimental consequences for biodiversity. So in Serbian area, we lost one subspecies. And we also detected clear signs of population admixture on all underlying genetic markers, and really weak signs of locally adapted population, which indicates that good part of genetic variability is lost. And that is worrying, especially in the light of the lost potential of populations to adapt to the changing environments. And population that are adapted to specific ecological and geographic influences and environments no longer exist. We concluded that this is mainly due to the human influence, and that humans really disturb natural distribution of honeybees subspecies in ecotypes, especially to the migration beekeeping, migrating of apiaries, import of the queens, and non-selective exchange of queens. But all our underlying genetic markers showed us two things. We can see genetic differences between managed and free-living honeybee colonies. And there is always a higher level of genetic diversity in free-living societies. They are always more diverse. So what these two things actually tell us is that free-living honeybees are important reservoir of genetic variability. They have those allelic variants that have a potential to help population to adapt to changing environments. And due to the fact that genetic pool of free-living colonies is shaped by natural selection, they retain more potential to adapt to if environment change, and they can definitely live on their own. So I think that in the native areas, it is highly important to preserve free-living honeybees colonies and to let them evolve on their own. And to do that is only way to do that is through habitat protection, preservation of native ecotypes and subspecies, and limitation of import of non-native subspecies, and also through sustainable management of commercial hives with special regards to carrying capacity of the environments. So for the question, are wild bees too? My strong belief is that honeybees were never truly domesticated, and that they are only kept. And as our project logo says, honeybees are born to be wild. And with that, I would like to thank you, my amazing team, Katarina, Slobodan, Pavla, and Alexandra, our funding agencies, and to you all for your attention. Thank you.
Thank you, Marija. So very interesting. And I think that the takeaway here is the importance for genetic variability, and that, bees that have evolved for thousands of years into these subspecies have, the greater ability to survive in their locations. And when you start introducing foreign bee genetics into an area, it really has implications. So thank you for contextualizing all of that. Very fascinating research.